Good afternoon. I'm Thomas Wachtler, and I'm chairing the session on FAIR approaches for electrophysiology at the INCF Assembly 2021. So when we're looking at the FAIR principles, at the first publication of the FAIR principles, this had a clear focus on data publication and hosting. In the sense that it was concerned with, like, for example, persistent identifiers, access protocols, and so forth. But there's also, and that's of course the important part when we think about making science open, making research available. But there's also other aspects that those that host the data and provide the data stewards, as they are called in the publication, that they can only fulfill once there are the prerequisites in the data that they get by the contributors, that is the researchers. For example, the formats that are used, for example, very importantly, the metadata that are provided. The descriptions and of workflows and provenance and so forth. So these are this is important contributions that need to be taken care by the researchers in order to provide the prerequisites for making data fair and having fair data. So there are these two communities or two stakeholders, both on one hand, the data stewards that are concerned with the kind of long-term provision of the data, and there are the contributors that provide the data to begin with. And we're going to look at these two perspectives, these two players in the game a little bit closer later in this session. So an important part or important requisite, of course, is that both roles need to play together, need to interoperate. So meaning that if there are data repositories that require certain requirements on the data, then researchers must fulfill them, and in order to make the data fair, in order to be able to provide fair data. And when we're looking at the field of electrophysiology, then we're concerned, or we are faced with this notorious challenge that researchers face in data sharing electrophysiology data, that lies in the on one hand heterogeneity of the data and also the complexity of the data, and the high variety both between labs and also even within a lab and within one experiment, where experimental paradigms are highly sophisticated and diverse kinds of data are acquired in a single experiment, even, and need to be kept in a way that they can be related to each other in a meaningful way. And also there's typically in electrophysiology a high amount of complex metadata that go with it, that need to be kept track of and need to be recorded in order to eventually lead to interoperable and reusable data. So the reusability part is one that lies very heavily on the data provision as given by the researchers. And so having this heterogeneity, one way to deal with it is to try to standardize, and that is something that luckily INCF is supporting. INCF is started to identify standards, community standards, and endorse them in order to help the researchers choose tools and to choose them such that they can make their research more efficient and can have an easier way of making that data according to the FAIR principles. INCF has already endorsed some standards even in the field of electrophysiology, but there's of course many more tools and services and de facto standards that are out in the community that researchers in electrophysiology need to employ and to utilize in order to make their data management efficient and in order to arrive at FAIR data. And so we have again a high variety of tools, and here again we have this challenge. I think that we'll have to deal with and have to find ways of bringing these tools and standards together so that they work together to achieve interoperability, so that tools that are made for different tasks in the data lifecycle, in the data processing, can work together in a meaningful way and in a way that actually makes the research efficient. And so we're going to look at hopefully, I think today, how some of these tools are being employed. On one hand, for data repositories, for data provision to the community. So we're going to look at 3 platforms for electrophysiological data, or that have focus on electrophysiological data. One is the EBRAINS platform that Andrew Davison is going to talk about. Then Yaroslav Halchenko will talk about the DANDI archive, and Tom Johnstone will cover the EDAPT platform in Australia. So we have 3 platforms on 3 different continents, actually, and we'll have a large focus on this level of FAIR that addresses the data provision to the community and long-term hosting. But then we'll also switch perspective and look at it from the bottom-up perspective, so to speak, with Sylvain Takerkart, who will talk about a recently established community approach to arrive at a standardized data organization for electrophysiology. And I would like to ask the speakers now in turn to take turns with their talks, and invite Andrew Davison to the first talk. Please, Andrew.
Thank you very much, Thomas. It's always difficult to figure out where the unmute button is when you're screen sharing. So if I can get back to my first slide. So hello, yeah, I'm Andrew Davison. I'm from the Paris-Saclay Institute of Neuroscience and the Human Brain Project. And I'm going to talk today about curating electrophysiology data specifically, and specifically for reuse in the context of the EBRAINS research infrastructure. Now, we've heard a lot about FAIR in this conference. We want data, computational models, and other research products to be easy to find, easy to access, easy to use, easy to combine with other data or models, and easy to reuse. And reusing models and data is in many ways the most challenging part of FAIR, in part because it builds on the first 3 attributes. So findable, accessible, interoperable. So these are prerequisites before we can even think about reusable. So in this talk, I'm going to focus on reuse, and specifically on the use of electrophysiology data. So there's an old joke in the field. An electrophysiologist would rather use another electrophysiologist's toothbrush than use their data. So why would we want to reuse electrophysiology data? aren't most data acquired to address a specific hypothesis, and aren't they therefore of little use outside the context of the original experiment? Now, while the first part of that is mostly true, the second part is much less so. There are at least 4 ways in which electrophysiology data can be reused. They can be reanalyzed to test new hypotheses not consulted by the scientists who originally obtained the data. They can be combined with other data and used for meta-analysis. They can be used to build computational models to inform the parameters of the model. And they can be used to validate computational models, to validate computational models to measure how well a model represents the biological reality. I don't have time to show examples of all of these, so I'll focus on one example of meta-analysis. And this example comes from a series of papers in the Journal of Neurophysiology from Srijoy Tripathi and his colleagues, based on their database, neuroelectro.org. Now, the major challenge of meta-analysis is ensuring the data from different studies are comparable. And given the general lack of standardization in neurophysiology, that's a major challenge. And it puts additional constraints on the metadata that are needed. So this figure from Tibekin et al. 2018 shows the resting membrane potential of hippocampus CA1 pyramidal neurons in blue and of striatum medium spiny neurons in red, measured with intracellular sharp or patch electrodes in brain slices across lots of different studies. So the error bars show the standard error across all the neurons recorded in each study. So we can see that the variability between studies is much larger than that within each individual study. Now, the intra-study variability presumably comes mostly from the intrinsic biological variability of the neuronal population. The variability between studies could be due to many things. So differences in recording conditions and protocols between different labs, differences in the precise population that's been recorded from, and or differences in data analysis. And so the neuroelectro studies were able to show that a lot of the variability does come from differences in recording conditions between different labs. So taking some other examples from one of these papers of neuronal parameters extracted from the data, we can see, for example, that the input resistance is consistently higher for patch than for sharp electrodes. We can see that the membrane time constant of these pyramidal cells, or perhaps this is all cell types, gets shorter as the animal ages. And we can see that the action potential threshold depends strongly on whether a liquid junction potential correction was applied or not. So using a regression analysis, Tripathi, Tibekin, their collaborators showed that the experimental conditions shown here, so in this list, these factors accounted for 43% of the inter-study variance in neuronal parameters. And they were able to use this analysis to normalize the parameters, in other words, to adjust the parameters to the values they would have had if recorded under a standard set of experimental conditions. And this normalization greatly reduced the variability seen between labs. However, that still left more than half of the variance unaccounted for. So for one thing, not every component of the bath and pipette solutions was included in the analysis, and nor with the details of how the slices were prepared. Equally, perhaps pyramidal neurons in different parts of CA1 have different properties, and maybe different labs recorded from different subregions. And then finally, it turns out that the protocols and analysis methods used to obtain the neuronal parameters from the raw data were also not standardized between labs. So this figure shows the different methods that were used to determine neuronal parameters across different articles. So taking panel B, for example, in the case of the input resistance, 6 different methods were used across 22 articles just to calculate the input resistance from the raw data. And in fact, about 20% of the articles didn't even report which methods they did use. So to sum up, even for this relatively simple scenario of measuring single neuron properties, a considerable amount of metadata is needed, covering details of the specimen, the precise recording conditions, the experimental protocol, and the data analysis methods. Now, we've seen some use cases for reuse of EVS data, and we've seen just how rich the metadata need to be for even fairly simple analyses. So how can scientists know which metadata are needed? Do we have standards for this? And how should we share our metadata with others? So in fact, there are several different projects that are currently trying to develop standards for which metadata should be recorded and how to represent them. This is, I think, a partial list. I suspect that the Spark and Dandy projects also have standards under development. So consider this to be a partial list. So first is Neurodata Without Borders, or NWB. You probably may well have heard of it. It's a standard format for neurophysiology based on HTF5, endorsed by INCF. One of the key features is that the detailed metadata are stored in the same files as the raw data. And if we look here, for example, at the NWB module for EFIS, intercellular EFIS, we can see space for all the recording conditions that we identified previously in the new electro example as being essential for this reuse. ODML is a file format for storing arbitrary metadata. It was created by the German neuroinformatics node, which also provides a number of standard metadata terminologies. And we see here, for example, an example of the suggested metadata for describing an electrode. Neuroshapes is a project to develop open schemas for FAIR neuroscience based on semantic web technologies, in particular the W3C PROV and Shackle standards. It has an INCF working group, and it provides very detailed metadata schemas based on a data provenance approach. OpenMinds is a set of metadata models for research projects in neuroscience. It's being developed primarily by the Human Brain Project for EBRAINS, but it's an open source project, and we are looking to grow the community. Like Neuroshapes, it's based on a linked data approach using JSON-LD, and it has a modular approach. So there are modules for basic metadata, for controlled vocabularies, and for spatial metadata already available, and there is an OpenMinds module for electrophysiology currently under development. And finally, many of you have probably heard about the BIDS family of standards, which started out in neuroimaging and now has extensions for various electrophysiology modalities, including EEG and intracranial EEG. And I won't say any more about this now because Sylvain will talk on this topic later in this session. So I've presented these as competing standards, but in fact, they are largely complementary. So for example, NWB is primarily concerned with representing metadata from a single recording session in a single file. Neuroshapes and OpenMinds are concerned with representing metadata in a database and with metadata exchange. And BIDS is about storing data and metadata from multiple subjects and recording sessions in a single bundle. I think an important task moving forwards is to maximize interoperability between these different tools. And I think just as a quick aside, the most promising way to do this is to use, again, semantic web and linked data technologies. So I don't have much time to go into this in detail, but the idea would be for each of the metadata models I just talked about to map the terms they use to ontologies, or at least controlled vocabularies using global identifiers. And to give an example, so JSON-LD is a very nice tool for this because it lets you use simple, meaningful names in the JSON document, like name or bath solution, then map them to full IRIs using the so-called context section. So here you can see that we map name to the schema.org definition of name, bath solution to the Neuroshapes definition, and then we can have cross-links between these. Okay, so now we've established the value of reusing EFIS data. We've established we need a lot of metadata for reuse, and we've established that more or less standard formats are available to define what metadata we need and how to store and exchange them. So the next question is, who is going to gather all of these metadata and put them into these standard formats? The person who's best placed to do so is the person who did the recording in the first place, who's probably a postdoc or a PhD student. But it can be a considerable amount of effort to do all of this. And with the current reward structure of science, the return on investment in terms of career progression is frankly not very large. So there are many things we could do to improve this situation, as funders, as hiring committees, and so on. I've already heard valuable discussion about this at this conference. But I'm going to focus now in the final part of my talk on one specific thing we can do, and that's providing professional curation support, which is what we're doing in the EBRAINS Data and Knowledge Services, which is funded by the Human Brain Project. I'll briefly at this point introduce EBRAINS. You may already have heard about it from other talks at this conference or at the EBRAINS booth. You're very welcome to visit the booth for more information. So EBRAINS at ebrains.eu is a new digital research infrastructure created by the Human Brain Project, funded by the EU, that gathers an extensive range of interoperable data, models, tools, and services for brain-related research. And among these is the eBRAINS curation service. So if you would like to share your data to the eBRAINS data repository, the curation team will accompany you through the process, including support for uploading the data to long-term storage, which is at the Swiss National Supercomputer Center in Lugano, support for choosing an appropriate license, and support for providing the necessary metadata. Now, an important feature of eBRAINS curation is what could be called progressive refinement. So I remember a presentation, I think at this conference, by Colin Ingram and Leslie Smith many years ago about the Carbon Project, which was one of the earliest efforts to build a large-scale data sharing portal for neuroscience. And they talked about how they had to reduce and then reduce and then reduce some more the amount of information they asked for on the first page of their metadata entry form, because otherwise it was just too intimidating. So you really had to slim down how much data you asked for at one time. And we take a similar approach in eBRAINS to make the process more manageable, both for the data provider and for the data curators, with a 3-tier curation process. So tier 1 curates basic metadata that are common to all neuroscience data sets. So are these data from humans or from animals? Which species, which strain? Which files came from which subjects and from which samples? What methods were used? Did the research have appropriate ethics approval? And so on. Tier 2 adds spatial metadata, from where in the brain were these data obtained. And this can use either precise stereotaxic coordinates, reference to standard atlases, or simply specify a brain region using standard ontologies. And then finally, tier 3 adds in-depth metadata. This is where we include most of the details necessary for reuse, such as the electrode properties, the bath and pipette solutions or patch clamp, the detailed stimulation protocols, and so on. And so you might ask, how's this going? So we have currently 1,036 data sets in the eBRAINS knowledge graph at the moment of this presentation, which have all undergone tier 1 curation. 94 of these are electrophysiology data sets, and of these, 39 have now undergone tier 3 curation. So a large part of this gap between tier 1 and tier 3 curation is due to the time required to develop the metadata schemas, so we will speed up. However, in-depth metadata curation is intrinsically a more time-consuming process than basic curation. So we're also working on ways to support data providers in the annotation of their own data sets, and working with existing efforts such as NWB. So for example, if we have a fully annotated data set in NWB format, we can automatically import this into the eBRAINS knowledge graph. We can import all the metadata that we need at tier 3 to reuse that data. And so you have the metadata only both in the file and in the database. So my talk is specifically on the topic of curation for reuse. And so you might ask, how can you, as someone interested in reusing a data set you find on eBRAINS, access these metadata? So unfortunately, I don't have time to give a demo here, but I can summarize the different tools we have and are developing. And if you're interested in learning more, please feel free to contact me after the presentation. So all metadata in eBRAINS, be they tier 1, 2, or 3, go into the knowledge graph. The knowledge graph has a REST API and a graphical search interface. At present, tier 3 metadata are only accessible by the REST API. We are working on adding these in-depth metadata to the GUI. We're also developing a higher level REST API specific for neural activity data, such as e-phase recordings, and an associated web app, which will provide a user interface focused more on the in-depth metadata. And then finally, we are beginning a project to convert all suitable eBRAINS datasets into standard formats, initially NWB and NIX. And this conversion should be largely or fully automatic based on the metadata stored in the knowledge graph and on the NEO library. So thank you. I'd like to thank you for your attention. I'm looking forward to the discussion. I'd also like to point, if you're interested in this topic more generally, the RDA has a working group on curation for reuse, which is working on this issue across all scholarly domains, not just neuroscience. And please visit us at ebrains.eu. Thank you.
Thank you, Andrew. And Jarek is the next speaker, please.
No, let me try to share my screen. Does it share my slides? Hello? I would assume that it shares well.
Yes, it does.
Oh, okay, good. So I'm here representing DANDI, which is Brain Initiative Archive for publishing and sharing cellular neurophysiology data. But it's not the effort which is of my own. We have multiple institutions involved. Satra is leading at MIT. I am participating from Dartmouth. KIT were helping developing a web platform. Catalyst Neuro with Ben Victor assists with data conversion, harmonization, and of course we collaborate with many initiatives out there and projects. And the whole project is supported by NIH. That's why it's Brain Initiative Data Archive. And also we've got support from Amazon Web Services. So all data is stored for free at the moment with funding provided by Amazon. So once again, DANDI is archived to store cellular and neurophysiology data. But as you might have seen this INCF figure, the whole spectrum of data is large, and even at cellular resolution, there is a wide range of modalities. And it might be behavioral data as well. And we also get into data sets which are multimodal. So typically now it's not only electrophysiology recordings, but it might be accompanied with some MRI recordings or staining. And that puts additional aspect on the archive, such as data is not only large, because some of the data might be in pretty much terabytes per single session, or it might be tiny, but also it's wide, right? So we have wide range of data which spans a big range of different instruments, protocols, and analysis. And with this data, of course, we want to assist driving developed models and also improve experimental designs going forward. So from the platform, we need to, from the archive, we want to provide infrastructure which would assist not only with dissemination and search, which would be kind of primary aspect for reuse of metadata, but also visualization and computation. And we see future of the archive not only as the dump of metadata or dump of data, but also which would be used for collaboration and coordination between different groups. And to achieve that, of course, we need to make it efficient and also fair. But what it entails, or how could we grasp such large and wide data, we need to work with standards. So in our case, at the moment, we are relying on two major standards, such as Neurodata Without Borders, or specifically MWB 2.0. And also we are starting to add support for BITS data, which is close to my heart because I come from neuroimaging domain. And to work on this, we don't want just to keep those two standards apart. We want to incorporate and work. We are working already with those groups and communities to integrate metadata organization and data organization within the two, and work with different BITS enhancement proposals to harmonize efforts, pretty much, not only the standards, but efforts of different communities, neuroimaging and electrophysiology communities and microscopy communities. And also we want to provide somewhat inspiration, but also linkage to other efforts aiming to provide standardized ontologies and represent metadata as linked data, such as neuroimaging data model. And to do that, or make it accessible to the users, we want to make harmonization of the data easier. So let's say one of our clients or DANDI client, which users need to use to upload data to the archive, the ultimate goal is that we would just give some collection of data files on the hard drive, and based on metadata, they would be organized in some consistent hierarchy. At the moment, this hierarchy is pretty much inspired by BITS, but not based on BITS, because work on supporting electrophysiology is still ongoing, but it already provides harmonized view across all the data sets which shared on DANDI, even though they might not be harmonized to the ultimate bits format. And then we provide validation. So users before uploading to archive could easily validate data, either it's conformed not only to NWB, but our extra schema, which we develop on top of those standards, to provide harmonized view of metadata, which is provided by the archive. And we based it on, we developed such model in Pydantic, so it becomes very readily applicable for validation of metadata straight in the Python code, but also we export such schema as a JSON schema from which we can and do provide convenient graphical user interface to the users so they could enter and modify the metadata on the website. So it makes this loop of metadata curation easier, and we are trying to stay away from curating it ourselves, but rather encourage and facilitate users to enter all necessary metadata within their DANDI sets before they upload. And also the ultimate goal would be to export all this metadata as linked data, so it could participate in the search in the larger linked data universe. And talking about metadata is great, and FAIR principles make really strong accent on metadata. But what is also quite important is the standardization and facilitation of data exchange itself. So this could be a schematic representation of how do we make DANDI archive data available to the users. First of all, DANDI archive itself, we are trying to develop it so in the future there could be multiple instances easily installed on institutional, let's say, infrastructure. So at the moment we run it in the Amazon Web Services and all data gets uploaded to Amazon, but also you could run it locally with simple Docker Compose recipe, which we provide, which would be using MinIO instead of Amazon S3 for local storage. So how users interact with the archive, they interact through before mentioned Python client to upload the data directly into Amazon S3. So our web front end pretty much just tries to stay away from any heavy traffic, and we just interact with the users and manipulate metadata while all the data interface directly into Amazon. And by making it available from S3 makes it quite convenient to the users to get the data, because they could either get it from the web interface or they could use our Python client, or we also expose all the data as data-led data sets. If you are into versioning of the data, I think that might be the most scalable solution at the moment. So all the data sets are available also on GitHub as data-led data sets, and while data itself still resides on Amazon Cloud Services. But then also, because it resides on the cloud services, we allow users to not even require download of the data locally, but they could run either their own Amazon EC2 instance, or they could use our Jupyter Hub, where all the data then conveniently would be available for download within the cloud. Or also there is work ongoing to make transparent access through ROS3 driver of HDF5, so you wouldn't even need to download any data which you don't particularly need for your analysis. Let's say if you just want to review metadata of specific NWB file, you could open it and just fetch all the blocks of data which necessary for that, which makes it much more efficient. And also we try to integrate archive or make archive easily integratable into other platforms. Let's say this is an example of NWB Explorer, because all the data comes through regular standard protocol to fulfill FAIR accessibility requirement. You could just copy that URL into the NWB Explorer and get access to it within NWB Explorer from MetaCell platform. Overall, DANDI archive is built on open source technologies, and we are trying to make it available or feed the use cases of many categories of users, right? So besides the data contributors, there could be citizen scientists who might want just to review the data or get access to it without maybe needing to convert any because they don't have any data of their own. But also we are trying to work with hardware developers such as OpenFS to make data collected in the standard form right from the hardware, because one of the major roadblocks at the moment is actually preparing the data in the standard form for the upload. Let's say all those NWB files to be created from a gazillion of manufacturer standards, not non-standards, proprietary standards, and then exposing data and metadata through all those components, which are again all open source. So on the right, you could find coordinates where to find more about DANDI. So to summarize how fair the DANDI is, I think we are quite findable at the moment in terms of that data could be found using simple queries on the web, and we are working on making metadata exported as linked data, so it could be participating in, let's say, dataset search of Google, and also provide our own graph server to make provide Sparkle queries search capability. Also, it's quite accessible. We are not creating new standards for any data containers. We are relying on already ongoing initiatives and standards, and also trying to expose metadata in standardized form so that it could be accessed by other services. And of course, we use just standard protocols, and there is a wide range of ways how to access data itself. We're trying to stay interoperable. We are using trying to use as much standard ontologies and vocabularies provided by already available instead of creating our own, and making sure that the data is reusable, of course, that license should be specified, and also that data is accompanied with rich metadata following the schema. And the last but not least, everything what we develop is open source. I mean, trying to develop it not just open source bits and pieces, which then would be difficult to deploy on institutional infrastructure, but rather that the whole project would be easily reusable, not only data or metadata which it provides. Oh, and there is a little bit of ontology. So what is a DANDI set? DANDI set is our way for naming data sets which are shared by DANDI, which come as a collection of assets and associated metadata. What is upcoming? We are finalizing development of our API server 1.0. So any minute, pretty much, migration is happening as of speaking. And then we will work on establishing publishing functionality. So we'll mint UIs for every release of a data set. Of course, further improvement of user interactions and user experience, integration with other applications out there already for, let's say, supporting NWB data. And as I mentioned, direct work with hardware manufacturers to assist with creating or collecting data in standard form straight from the hardware. And I think that would be it for DANDI for today.
Thank you very much, Jarek. And the next speaker is Tom Johnstone. Please, Tom.
Hello, thank you, Thomas. I'll just see if I can share my screen here now. Let's see. Is that working?
Yes.
Great. Okay. Thank you for inviting me to this session. What I'd like to do is describe a brand new project that we're just starting really in Australia that we've called the Australian Electrophysiology Data Analytics Platform. And this is something that was funded by the Australian Research Data Commons, which is sort of a federal level, national government supported infrastructure facility across Australia for research informatics. And what they were looking for were FAIR platforms. And so this is our FAIR platform that we will be developing over the next 3 years or so. So this is really a plan that I'm going to present you with, although we've already made some progress building upon previous technologies. So just a lot of text here. I'm sorry about that. Well, what I'm talking about here when I say electrophysiology, I know it means different things to different people, but really we're talking about something very broad. We're talking about anything that relates to recording neuronal cortical activity in some sense, typically electrically, but maybe using other techniques as well. We include magnetoencephalography in this case as well. And this was driven by the fact that in Australia, at least, there's no real advanced analysis solution in Australia for people performing research that uses any of these techniques. And research groups are extremely diverse. So they consist of people working in neuroscience labs, in animal labs, in human neuroscience labs, all the way through to clinical settings in the medical fields, as well as AI, engineering, and even things such as social and sports psychology, where they're increasingly using scalp-based electrocortical measures, EEG, and so forth. So the diversity of people doing these sorts of analyses or collecting this sort of data means that they've equally got a huge diversity in the way they go about doing this. And we have a large number of users who really are very unfamiliar with advanced computing of any sort. Many of them are unfamiliar with Linux or any sort of Unix-based system. Many of them have limited or no experience and perhaps limited access to supercomputers or high-performance computing facilities. And many of them are locked into proprietary hardware-based software that comes with the instruments that they purchase to collect the data, or perhaps idiosyncratic local analysis code that gets passed down from one postdoc or PhD student to the next. And so viewing this, we saw, and we did this through a number of community consultations over the previous year, 2019-20. We realized that there was a lot of open source analysis software in the form of packages that really could help people in these other settings advance their field and produce data analytics, data pipelines, and also data as an output that was more fair, that was especially more reproducible, which is the primary focus at the outset. But also, we wanted to make these sorts of analyses far, far more accessible to those types of users. So that was the key. How do we make that happen in Australia in an easy, accessible way and a portable way? Because we have labs in pretty isolated places with fairly basic facilities. And we want to be able to get these analytic tools to those users where they actually are. So our approach really was to create a data analysis platform that was built around structured data that conform to FAIR principles from the outset. So rather than getting people trying to put their data into FAIR or satisfy FAIR principles once they've collected and analysed it, we wanted to get them started on a platform that introduced those principles and gave them easy ways of making their data fair, accessible, and reproducible. As I said previously, we have a particular focus on making whatever tools that we create here very accessible to regional universities, small-scale labs, and labs with less technical staff. Right. So really the basis of our analytics platform at the moment, the plan for it at least, and the way that we started to construct it is to start off with a very user-friendly desktop environment for electrophysiology, broadly defined, as I said, related behavioral and psychophysiological data analysis and processing. And this is all based around containers, and I'll go into a little bit more detail about how we're constructing this. We have a desktop container that actually runs on Docker and provides a uniform desktop environment, a Linux-based desktop environment for every user, and that can be run equally on a Windows PC, a Mac, a workstation, a virtual machine in the cloud, or a high-performance computer. We have continuous integration builders for automated building testing, uploading and downloading and installing containers to this desktop environment. And those containers will be either software packages themselves or data analytic pipelines that have been created by the researchers, or the combination of data analytic pipelines and associated data and metadata. So really encapsulating their entire analysis process in a form that can be reproduced by other researchers using exactly the same platform. We realized to get people to do this, we're going to have to give them integrated tools to get them off or away from their idiosyncratic electrophysiology data formats, metadata formats, into something more standard. We've chosen BIDS as the primary standard, but we're very open to others, and listening to some of the other talks in this session, I think we'll have to be. We're basing it, or we're integrating it tightly with DataLad to provide data and code provenance, and do so in a way where people don't have to bother too much about the details. So we want this really to be operating under the hood, rather than the users themselves having to understand the ins and outs of DataLad or Git repositories. A key part of this platform was to make it interoperable with especially two other platform projects going on in Australia and funded by the Australian Research Data Commons. That's the Australian Imaging Service, which is a web-based XNAT database for image storage, typically image storage, and analysis pipelines for analysing those images. That's not exclusively biomedical imaging, but that's a large part of it. So what we're going to do is add electrophysiology pipelines into the Australian Image Service, extend the XNAT format to cater for electrophysiology or related types of data storage formats, and at the same time provide an easy interface within our desktop environment so people can go back and forth between the Australian Image Service and the AEDAPT platform. And likewise, we're working with the Australian Characterisation Commons at scale, which really focuses on data provenance from the outset, from the moment the data is collected from the instruments, gathering the data from metadata from the instruments, and also following that data all the way through the analysis pipeline. Clearly, we'd like to integrate, and we've already spoken to the developers of BrainLife, and we'd like to integrate as much as possible with these other platforms. Open Neuro is an obvious case, and I think Neurodata Without Borders also. This whole project is meant to be community-led, and we have some influence, well, we have not influence, we have people involved in the Australian Brain Alliance and the International Brain Initiative involved in these sorts of data standards that we've been talking about in this session. So really the primary aspect of this is to try to build something that's as easy as possible for people to use. And by people, I typically mean people who probably won't know what a virtual machine is. They probably won't really know what a Git repository is. And they don't really want to know about any of that. So we want to make it very easy for them to install and use these analysis packages and share their data pipelines, their data analytic pipelines, and their data in a format that can be understood and reproduced by other researchers. So our starting point was to create a standardized desktop environment where we have software installed, a wide range of software packages pre-installed, and installed in such a way that we avoid sort of conflicts and so forth in dependencies. we've put the entire working environment within a container. And the advantage of that is that this container can run on various platforms, as I've mentioned. And the nice thing about it is that it's going to look and operate the same way for the user, regardless of what platform they're using it on. And we see this as an important way of easing the transition from people, for people to go from desktop computing through to cloud-based computing and high-performance computing, if we can provide them with a very consistent environment. But it should also allow them to reproduce their analysis on the different platforms without really having to change much, if anything, in terms of their pipelines or code. So our starting point here is a virtual neuro machine. This is a container of a Linux desktop environment. And it's installable on a computer with Docker. So you have to install Docker, for example, but then it's basically a download and you run Docker and the whole thing pops up and you're ready to go. It doesn't even require a VNC client or anything like that. You can run it straight in a web browser and have a desktop environment in your web browser. And in fact, you can install this on I've done this at my university on highly, highly protected desktop computers in computer labs where we're not meant to be able to install anything. And I got this up and running in about 5 or 10 minutes without any problems. Below that, we have something that's based upon NeuroDesk, which is an already created environment, or there's a lot of work that's already been done on this. NeuroDesk is a desktop environment which has all these has a base working environment and containerized software. A huge range of different software from neuroimaging, and now we're extending that with a range of electrophysiology related software. So you can see just a few here, and you can see we're starting to add at the bottom in terms of the software for any sort of electrophysiology, EEG, MEG, and so forth analysis. Move on. So part of the reason for doing this, the philosophy behind it, I guess, is that, as I said, we want people to be able to transition from doing things on a local desktop to all the way up through high performance computing. And we want to be able to do it in an environment which is identical or as close to identical as possible when they're going from one platform to the next. And I think key to this as well is that we're going to be training these users and providing online tutorials as part of the platform. So we want to be able to guide people through this entire process from beginning to end with these tutorials. And the platform should be suitable as well for creating online workshops and scalable workshops for conferences and so forth. And finally, the justification here is we want to be able to, as I said, we want to be able to share the working version of a data and a pipeline, and we want that to be basically a click of a button from the user. So automated containerization of their data pipeline and data and metadata, so that they can easily share it with other users, or indeed bring it to a different computational platform, install the AEDAPT platform on that platform, and then run exactly the same analysis as previously. We have continuous integration, container and automated container building. I can't talk much about this at all, but Stefan Bollman is really the brains behind integrating this with the AEDAPT platform. This is a sort of a schematic of where we're at in the platform at the moment. So what we see, I don't know if you can see my mouse. In the left-hand column, we have users who wish to do computationally intensive, small and large data set batch processing. And here we see integration with high-performance computing with the Australian Imaging Service, which is based upon an XNAT database with web-based pipelines. BrainLife.io, which is a similar sort of web-based environment for automated pipeline processing, and any other platforms that perform that sort of analysis. And that is being developed. The NeuroDesk in the center column here is being developed to integrate directly with that and push pipelines, push containers for analysis pipelines up to, for example, the Australian Imaging Service, where they can be applied to data there, but also pull data and pipelines down onto NeuroDesk. So it's really you can start at either end, basically. We have the core of AEDAPT is the green column in the middle. So we have the container registry, which contains all the standard containers that we will provide for the different types of software and software packages, but would also be a place where users can upload their own pipeline containers. And we have this lightweight Linux desktop virtual neuro machine, which runs as a container under Docker and allows you to gives you a standardized user interface across different platforms. And then on the right-hand side there, we have integration with virtual machines, GPU-enabled virtual machines, and a Linux desktop environment that runs directly on high-performance computing system, the CDL. This is a schema for how the platform would look, but the key thing here is that it's built upon standards such as BIDS. It's got DataLad integrated tightly to allow for data and metadata tracking and provenance. And we're hoping to integrate as much as possible with the other initiatives such as the INCF, the International Brain Initiative, and the Australian Research Data Commons own initiatives in metadata standardization and data standardization. And I'll mention one thing here, and that is the Australian Research Data Commons has its own Research Data Australia service, which is a repository of all, not of all, in principle, it could be a repository of all research data collected in Australia. So we want to make it as easy as possible for people to integrate or upload their data and metadata into the ARDC Research Data Australia research repository. And the ARDC also has a service called Research Vocabularies Australia, which allows people to create and extend and modify research vocabularies from particular domains. And we intend to create some research vocabularies there and upload them, but would like to do so as much as possible with the INCF and International Brain Initiative. And I think that's probably where I'll stop. As I said, this is really, we're just getting started at the moment. We've got a prototype system that we're adding to. We're going to have a hacking session at the OHBM conference in June. And we're very happy to invite any other people who want to be involved to get involved. Thank you very much.
Thank you, Tom. And the last talk will be by Sylvain. Sylvain, please.
Hi, everyone. Let me share my slides. All right. All right, so I'm Sylvain Takerkart, working at the Institut de Neurosciences de la Timone in France. It's a 150 people neuroscience interdisciplinary institute located in Marseille in the south of France on the Mediterranean coast. So today I'm going to talk to you about a standardization initiative that we're pushing at the moment, which is dedicated to try to organize the data of electrophysiological recordings obtained in animal models. So my talk's going to be in three parts, and I'm first going to describe a little bit where we're coming from at INT, which is a project-specific verification environment that was put together during a few years. Why? Because of a collaboration that was starting between two groups. The group at INT in Marseille of Thomas Brochet and Alex Harile, and the group of Sonia Grun in the Jülich Research Center. And to summarize, the experimental lab was the one in Marseille, where we were performing data acquisition, electrophysiological data on awake behaving monkeys, while the group of Sonia was the analytics group, where advanced data processing and modeling was performed. So there was an obvious need of exchanging the data in one direction, but also in the other direction. And so what was required was to assemble data and metadata. So this is a classical thing that we've been talking about. But let's look a little bit at it. So this is the setup for the particular experiment that we focused on during this project. A monkey was implanted with a Utah array, and his activity was recorded while performing a visual motor task. And to perform data analysis, we needed to put together lots of information coming from lots of different sources. And looking at it, there was lots of files and lots of file formats. And if you look even more closely at it, you realize that the challenge might actually lie more in the metadata than in the data itself. Okay, so complex task, and a complex task, fairly complex solution, but solution there was after a good amount of work that was built on top of a bunch of tools and components that you see listed here, and notably the Open Metadata Markup Language, or DML, which allows us to gather all the metadata coming from multi different sources into a single structure. So this solution was allowed us to fulfill the goal of what we were trying to achieve, which is data exchange between the two groups. Obviously, this work also meant that the data became fully fair, and we were able to share the data with the wider community, which is what we did. And this resulted in the publication of one of the data sets, which was a first for such data set. Okay, but this was project specific. So the next step, obviously, are to make these tools more generic, project agnostic. And for this, we cannot work alone. So we need to try to work in a community-driven manner to gather all the forces and also to make the tools as generic as possible. So for this, we got a bit of funding at INT. And the first thing that we did was actually to try to bring people together, to get them to discuss and to put them together. And this led to the creation of a new INCF working group, which is dedicated to the organization of data and metadata, but dedicated to animal data, because there was a lot of solution already available for human data, as you know, with BIDS. So we started last year discussing regularly, and now the group meets regularly, and we got a good number of active members. So it's a lively group, and we're making progress, even if the task is difficult. Okay, so now what we came up with, which is not, it's work in progress, of course. So lots of discussions at the beginning was about what existed in other fields or related fields, and obviously one of them is BIDS, the brain imaging data structure, which, as you know, probably is a simple and intuitive way to describe neural data. At the beginning, it was neuroimaging data, because it started with MRI in 2015. It's been extended a lot to other data modalities and electrophysiological modalities, but mostly in humans. And there are lots more extension under development at the moment. So the question that we had in the group and we were discussing with the electrophysiologists was simply to BIDS or not to BIDS. And there was a lot of debate around this because it's not very easy. BIDS doesn't really correspond to the habits of lots of electrophysiologists working with animal models. And you see here, I'm going to show you in the following slides, a few examples of data organization that are used in different labs. So here, this was the organization we were putting together at our institute locally, with something that looks a little bit like BIDS, but with differences. Here you have another organization in a different institute with the experimenter at the top level. Here another one where you have the modality at the top level, then the stage of processing just below, etc., etc. So there's a lot of diversity, but when you look at all these things, obviously all the concepts that you use that are used are the same. You have the subject, so the animal, the session, the modality, etc., etc. And all these concepts exist in BEADS also. So what we thought at some point is, okay, let's give it a go and let's try to see whether we could work with BEADS by extending it to handle electrophysiological data recorded in animal models. And so this is what I'm going to describe now. So this is also described in the poster. So very quickly, of course. So this is what a BEADS animal electrophysiology dataset look like. So you recognize, if you know BEADS, the classical hierarchy where you have the data sets, the subjects, the sessions, the modality, and the data files at the bottom of the tree. We're following, obviously, the naming rules, and we had to make choices on the data formats that we're supposed to, that we want to support. We need to support data formats that are both open and more or less standard. And for this, in electrophysiology, we have two INCF standards that are available, which are NICS and NWB, and this is what we're proposing at the moment. So you see here, it's a NICS file that's lying here. So for the metadata, you have some amount of metadata that's available in the name of the files and directories, but then most of it is available, is inside the sidecar CSV and JSON files. Either some of them are generic to all BEADS modalities, some of them are specific to the modality we're working with or we're trying to add. So the generic files describe the list, the animals themselves, or the subjects or participants, the general description of the data set itself, the task performed, etc., etc. And the specific files here where this is part of our work, and which is try to propose a description of the probes, the contacts, and the channels that are used to record and that are available in the data files. And then we have another file where we can put everything else. You have to put it in a structured manner. You have to put it by respecting some rules, but you have the possibility to add whatever you want. Okay, so that's what we're proposing, but and it's still ongoing work, and it's not part of BEADS yet. So it's we are still working on improving it, and we want the feedback from the community. But to get a chance to have such a standard adopted by the community, we need to have tools around it. And so I'm going to describe some of the tools that we're trying to develop as a group around BEADS animal EFIs. So first, and it's a very important one, and it's a project we have internally at the INT. We're trying to put together a system that allows going directly from the acquisition to BEADS for animal electrophysiology. And here the challenge, well, there's two challenges, the file format conversions and the metadata collection. And so for the file format conversion, we're relying on the NEO package. For the metadata collection, we are putting together a system that is trying to standardize the metadata that is collecting during the experiment. And this is the system we're proposing is both modular and version control, so that we can do things as cleanly as possible. And then we have a second piece of software which actually integrates the data and the metadata to end up with the desired BEADS structure with all the metadata. So this is a project by myself, Julia Sprenger, and Jeremy Garcia that you can see on another poster. So this is very important to us, and to provide such tools that will ease the life of the experimentalists. If you don't have that, no chance you're going to get adopted. So this is a key piece of the puzzle. Then we want to ensure interoperability, and I'm going to launch a bunch of names here that you've heard about during the conference and during the session. Why? Because there are like initiatives that are very close to this also. They're trying to either have a data model for electrophysiology, or the DANDI archive, or the eBRAINS initiative, or the Spark data structure. And what's interesting is that we actually have stakeholders of all these projects within our working group. So we are making sure, as you've already heard in the previous talk, that we are interoperable between these systems. Okay. Next interoperability level is with other data modalities, and here this is one of the large benefits that BEADS going to bring to animal electrophysiology, if all this works, of course. It's to be able to interoperate with other data modalities, because you're going to have all kinds of data in a single structure. So I'll give you two examples. When you do electrophysiology on human primates, now very often you do an MRI before recording the data on your animal, and obviously MRI is well supported by BEADS. It's been supported by BEADS since the beginning, so you're going to be able to have all your data and to get them to interact very easily in the same structure. Another example is that so one of the challenges here is that is to get proper support of animal data within BEADS, and we're sharing this problematic in what we're doing with another extension proposal of BEADS, which is dedicated to microscopy. And so we're working together with the people leading the microscopy extension proposal and with the BEADS community to do this as well as possible. Okay, but next, obviously, what we want to do is facilitate data processing. This is the obvious end goal of everyone, obviously. And so we have a few examples here that things we are starting. So we're working with a team of Spike Interface, and namely Samuel Garcia, to provide an interface between BEADS and so that a BEADS electrophysiological data set can be processed in Spike Interface directly. And we have also put together, Samuel Garcia and Julia Sprenger, a unified interface that describes the probe layout, which is also a piece of the puzzle. We are also discussing with the people from the Elephant Analysis Toolkit to see what we can do together. Interestingly, what BEADS adds here is the possibility to handle multiple sessions of data, while for now in Elephant, like it's been described in NWB also for the data storage, it's focused on single sessions. So it's really adding the multi-level session that might be the added value here. And we hope to have more to come. Okay, so take-home message. There's just one single take-home message, very simple one. If all this works, a big benefit is that we're going to construct FAIR electrophysiological data. So this is really the benefit, and we hope that the electrophysiological community will adhere to this proposal, because it will help the community in this direction. But we're just at the beginning, so obviously we need help. So get involved, join the working group. We have fun discussions. Just comment on the proposal itself, or propose your data set if you have one and you see, oh, hey, BEADS sounds fun, so let's go for it. And if you're a software developer of tools that touches electrophysiological data recorded in animals, just get in touch if you want to see what we could do together. All right. This is the end. Thank you, everyone. Thanks for your attention.
Thanks very much, Sylvain, and thanks to all the speakers for the very interesting talks. I think we have a couple of questions that came in from the chat. And I would like to start with a very specific one from Nick Günther, who is asking Tom, what is the oldest analysis you have reproduced using Docker?
Yeah, so it's a really good question, and I gave a rather vague answer that not that old. I think the problem we've had, and many of our researchers have had, is that Docker has undergone so many changes and versions and so forth, and each one has a tendency to break earlier containers. They just don't run properly, and so the whole idea of Docker seems to be undone sometimes by the fact that it changes. And usually it's actually, though, in my experience, in our experience, is the way that the containers interface or operate with the environment that they're in that's changing, rather than the actual analysis code within the containers. That's the problem. So, you know, one solution to that is you just save the entire environment. It's not a very sophisticated solution, though, and I think there have to be better ways of solving that problem. Stefan Bollman is our expert on containers and the whole containerization aspect of this. I'm sure he'd have a lot more to say on it. I'm afraid I can't say much more than that. Oh, everyone else is on.
Sorry, I was muted. Yeah, I guess that also alludes to maybe a larger problem in principle, that even standards kind of can change over time. So how do we deal with that? Is it should we try to make sure that all our infrastructure is somehow flexible enough to be adapted also to changing standards and changing conditions? Well, what are your thoughts on that?
Could I
I mean, look,
Jarek was sorry.
Oh,
Jarek.
yes, please, Jarek, please. Yeah.
Sorry for the previous point. So Docker containers, how much? It's not only about reproducing as, oh, you take container, you rerun it, you've got the same result. Containers also help to isolate your environment, right? So whatever result you achieve, if you know that you ran it in container, you have better chance that you had all necessary components inside and no outside environment inflicted upon it. So even if you wouldn't be able to run that container, you would have better idea that, oh, it's more likely to be reproducible. That's one. Second, even if you cannot run the container, you should be able to introspect it. So you could look inside and see what versions were used, right? And then maybe based on that knowledge, if container doesn't run, you could try to recreate container maybe with the current base, but those versions taken from, let's say, snapshots of Debian.org or newer Debian snapshots. That's what I was trying to do actually in the background quickly to demonstrate our paper from 2011, I believe, but I've run out of time. So it's not about reproducibility as turnkey, but reproducibility as encapsulating all the information relevant.
What about changing standards? I'd like to bring up this this question again, because if we look at bits has changed over time, right? So how do any approaches that rely on standards deal with that? Jarek, again?
There is a wide range of scenarios which is happening. In theory or in idealistic world, standards should be adhering to semantic versioning. So whenever you adjust the standard, previous versions should still be legit. Let's say you have a data set with version 100 of bits, right? You run bits validator, it says it's legit, right? And any tools which support series one of bits should be able to support that data set. But that's idealistic environment because we are still, boiling things down, right? And the same happens in NWB. There is ongoing work already in, let's say, bits to create a schema for bits. then at least validators could use schema to validate either data set is coherent with that version which it uses, which is not the case now, right? Because it would use most recent bits version to validate. And the same happened in NWB, right? Schema started to be embedded in the file, so you could, in principle, validate against the schema in the file. So overall, I would say in the future, SIC standards, they should be involving, they should be backward compatible. Any breakage should introduce major boost, for which there should be clear guidelines how to upgrade. And that is no how different from software, right? So APIs change, they adhere to the versions. So we just need to grow into the, just mature, I would say. And I think the future is bright.
Sylvain?
Yeah, just to complement a little bit with the BIDS example, and Yarik can tell more about it than me, but I will start. BIDS has been changing, but it's not. There's no backward compatibility that has been introduced, so it's not really changing. It's mostly growing. And this is exactly what we are facing at the moment with trying to handle animal data in BIDS, is that we need to introduce more concepts to somehow make it possible to handle animal data through BIDS as it is defined now by just adding some new concepts.
I have a quick question actually on that, Sylvain, because I'm wondering whether, because it's, as we say, you tend to think of these things as adding new functionality, adding new capability and so forth onto something. But have you encountered any aspects where you say, well, look, that aspect of BIDS, that standard of BIDS is just not compatible. It's not compatible with what we need to do, and it can't necessarily be solved by adding something new. I mean, are there aspects to BIDS, the BIDS standard, for example, that you see that you would like changed because it gets in the way or is an impediment to bringing in the animal data, the animal EFIS data?
Well, here again, Yarik is more involved in BIDS himself than me, but I can try to give a little bit of an answer. For animal electrophysiology, there's been this huge debate, as I described in my talk. So, okay, BIDS is not for us. Fair answer. So, but you say, hey, come on, look at it. You got your session, you got your subject. What's the change when you're going to have your script and your session is going to be on top of your subject or whatever? So the goal is to somehow automatize a lot of things on top of these standards. So it's true that we want to maintain as much as possible human readability and easy readability of everything. And this is what made the success of BIDS also at the very beginning, is that, okay, you see the structure, it's intuitive. So we need to maintain it intuitive. But at the end, what I'm going to say, some people are not going to like it, but some details like, okay, putting this level on top of this level, it's just a detail that doesn't really matter much. It breaks the habits of some labs. And maybe what the difficulty here for animal electrophysiology is that it might break the habits of a majority of labs, because the habits are very, very, very different and the practices are very, very, very different. But what we're proposing is really not very, it's not very different. So
Andrew?
Sorry, I was just going to say, I think the toothbrush joke is quite relevant here.
And Yarik has a comment.
Sacrifices should be made. If you look even to original BIDS kind of agenda, right? We want to account for 80% of the cases with 20% of the effort, right? So Pareto principle. Yes, some use cases might be a little bit disregarded for now. And that's how BIDS evolved, right? And that's how it involved in neuroimaging. Of course, bringing electrophys data from completely different domain sounds like a really difficult task. But as Sylvain mentioned, if you look at them, everybody uses pretty much the same in terms of directories, right? The directories describe some level as the first one, either it's subject, or now we have debate, oh, should we allow for other levels, maybe tissue, right? So what I see is bits would evolve to account for those use cases. Some difficulties should be resolved. Some use cases might not be accounted right away, but as it progresses, they will be accounted for, I would say.
Right. Let me, in the interest of time, let me move to another question we have from the audience. Matthew Abrams asks, and again, I guess that's for Tom, are the ARDC research vocabularies registered with NIF?
Yeah, I'm not sure whether they currently are or not. Through the Australian Brain Alliance and our involvement with the International Brain Initiative, I think we'd like to sort of broker that sort of arrangement to make sure that there is an alignment between what the ARDC is doing in that space and for example, the NIF. So I think that would make sense. And I think the 3 platform projects that I mentioned, ours and the other 2, are all on board with that sort of idea. So hopefully we'll see some progress there. Yeah.
This is something I'd also like to ask Andrew. How about EBRAINS, the metadata vocabularies in EBRAINS?
So in fact, I saw that question was coming up, so I was just Googling it to see if we are yet. So let's say if not yet, then soon.
Right. I think I don't see another question here, but I actually do have one for Sylvain. Because on one of your slides, you kind of mentioned you're in touch and want to be interoperable with DANDI and eBRAINS and other platforms. So the question is, how hard is that? Are the requirements compatible, or do you run into problems? And that again kind of refers to or points back to these platforms. How interoperable are these platforms? I think that would be an issue that we should be concerned about for the future, right? Yeah,
here's a challenge, as in most of these questions are more about the metadata than the data. Because sometimes you don't have all the metadata required by one system in the other system. So then what do you do? But other than that, it's mostly a matter of writing wire conversion tools between or importers or exporters from to a system to another. So it's every time it's a challenge, but we're just starting. So so it's not. I think it's not. It's possible to do it, but it's going to take time. It's important because we really believe that not all users will follow one workflow, but there's going to be lots of workflows, and at some point they will interact, and so we need really to be able to go back and forth.
I'm aware this is something. This is something which the IBI, the International Brain Initiative, is working on, and I think the INCF knowledge space has already done some kind of proofs of concepts for this, so they're able to pull data sets from various, of the worldwide resources and display them in a single search interface. So this is coming. I guess there's an IBI session later in the week, so perhaps people could turn up there if they want to know more.
Okay, thank you. So I was notified that we have a pretty strict time limit, so unfortunately our time is up. I would like to thank you again for your very nice talks, for the discussions, and also for the audience outside there for listening in. Thank you. Goodbye.